Let me make sure that that's sharing. That is actually a carbohydrate slide, but kind of pick up where we left off a little bit with reminding ourselves what we had done. So we had gone through starches and saccharides, and the monosaccharides being the monomeric or very basic unit of all of our starches. And so here we see some 6-carbon ring of sugars, glucose, another 6-carbon ring, galactose, and a 5-carbon ring of fructose. And you can join all these molecules together in a condensation reaction, also known as a dehydration synthesis, linking one sugar molecule to another sugar molecule to another sugar molecule, which is what you have here. Glucose linking to fructose through a dehydration synthesis in this direction. And then you have sucrose, which is a glucose plus a fructose and the byproduct of water. And you do these reactions over and over again, and you can get eventually glucose linked to glucose linked to glucose linked to glucose, and you can have that n many times, all right? When glucose links to glucose links to glucose, we get starch, and starch actually branches off in different directions. We could start branching our molecule going down that way, and there's a branching enzyme, and for getting rid of the molecules of glucose off of that, there's also a debranching enzyme. So it just makes it a nice compact storage form for glucose in our bodies. Right, so then we started talking about the lipids. The lipids are going to include the triglycerides, which are fats. They're primarily the fats that are stored in our body. This is the form in which we store fats. Phospholipids, which make up our cell membranes, and the steroids, which are necessary for a lot of molecules that we use, things like your steroid hormones, but also some other sorts of inflammatory molecules like the eicosanoids that we will encounter. Lipids, as I said, are the primary components of the cell membrane, okay? They're made of carbon, hydrogen, and oxygen, and they are nonpolar, and they're insoluble in water. They don't mix with water even. You kind of try to shake it up and mix it up, and the lipid will just coalesce right back together and float on top of the water because they are less dense than water. The most abundant lipids are going to be our triglycerides, okay? And these are used as our source of cellular energy. They contain more energy per gram of lipid or triglyceride than carbohydrates. The lipids will also have carbon, hydrogen, and oxygen, but they have far less oxygen content than do our carbohydrates. And we'll see that they will consist of, primarily when we're talking about the triglycerides, one molecule of glyceride to which 3 fatty acids are going to be attached. The phospholipids are constructed somewhat similarly to triglycerides, except that they have glycerol up at the top of their head and 2 fatty acids along with a phosphate group. We'll take a look at all of these. All right, so when we're looking at fats or triglycerides, we're going to have a glycerol molecule. And glycerol is just a carbon surrounded by 2 hydrogens and a hydroxyl group on each of the carbons. And we'll lose that hydroxyl with one of the we're going to lose actually the hydroxyl on the fatty acid and strip one of the hydroxyls of its hydrogen when we do our condensation reaction to make the triglyceride from the fatty acid and glycerol components. Now, last time we talked a little bit about these fatty acid chains right here. Whoops, I switched my mice. So hopefully my mouse won't be as trigger happy. Here we're looking at a big long fatty acid chain that is saturated. Every carbon is surrounded by hydrogen. It's a long hydrocarbon chain. We say it's saturated then, all right? And you could count your carbons here. And I think it says over here that this is palmitic oil, which is a 15 chain carbon. So this is getting kind of from the medium moving over into the long fatty acids. Sometimes those of you who are interested in nutrition, you'll read the differences of short fatty acid chains, medium fatty acid chains, the MCTs, oils, and the longer chain fatty acids. And there supposedly is some kind of special health benefit for medium chain fatty acids, okay? Helping with some inflammatory processes, a little bit better for people with Alzheimer's and epileptic seizures. I haven't ever looked at the data, so I don't know how true that is, but I do know that that's one of the common things that is looked at, all right? All right, so when we link our fatty acids to our triglycerides, we link to every carbon in the glycerol. So we can have like that long palmitic acid right here that's completely saturated with its 15 carbons. At the carbon here in the middle, we see we have an even longer saturated fatty acid, about 17 carbons, all right? And then over here on our first carbon of the glycerol, we have a double bond. Throws a little curve literally in the attached fatty acid. And this is in the cis configuration when we have everything on a double bond that's on the same side. We call it the cis configuration. If this H up here were over on this side and this H on this side, it would be the trans configuration. I'll try to draw that out in a second. All right. So this is an unsaturated fatty acid. It has a double bond. Not all your carbons are completely surrounded by hydrogen. And so this gives us oleic acid, an unsaturated fatty acid. And this can usually confer some level of fluidity. Your saturated fats are almost always solid, okay, at room temperature, whereas when we start putting in the double bonds, they become a little bit more liquid. The other thing, when somebody, I forgot who it was, asked about coconut oil, even if coconut oil has all saturated fats off of here, one of the differences with coconut oil that you find is they have a lot more of these medium chain fatty acids. So fatty acids that are in length of like 10 to 12 carbons. And especially if they take the fatty acids for the coconut oil, you know, that when they're bottling it to sell it, what they'll do is they'll warm it all up so it's liquid. And then as it comes back and cools, the parts of the coconut oil that cools fast, they'll skim that off. That tends to be something like lauric acid that's 16 carbons long. And that way they have a coconut oil that is predominantly the MCT oils, the medium chain coconut oils, and those tend to have a melting point and boiling point that keeps it liquid at shelf temperatures. So not so much that they put double bonds into it. They don't introduce that chemistry. They're just skimming off the long, heavier chains that would be more solid at room temperature. Okay. And you can pay a whole bunch of extra money for that MCT type coconut oil. All right, so cis and trans bonds. Cis just means the atoms are on the same side of the bond. So if I drew, you know, like we're drawing cis here, we have carbon going to carbon. And if the hydrogens were on the same side of the double bond, that is cis conformation. If I drew the carbons and we had the hydrogens on opposite sides of the bonds, okay, that's the trans conformation. All right. Okay. All right, so saturated fatty acids tend to have only carbon-carbon bonds, okay? And they're all saturated, all right? They're solids at room temperature. There are a lot of, more typically, animal products like butter, cream, okay? Unsaturated fatty acids have one or more, quite frequently more, carbon-carbon double bonds. So you can see these here. Oops. Darn it. Didn't have the draw on. So you see the carbon-carbon double bonds right here. Doesn't want to let me draw. And right here, okay? Most of these are liquid at room temperature and most are of plant origin. Now, if I look at this carbon chain on this fatty acid over here, and I count 1, 2, 3, 4, 5, 6, and I have a double bond right here at the 6th carbon from the end, that's telling me I have a 6 omega fatty acid, okay? A 6 omega fatty acid. We'll look at that on the next slide. All right, here from the end, if I count my carbons and I say 1 carbon, 2 carbon, 3 carbon, double bond happening at the 3rd carbon, this is a omega-3 fatty acid right in here, okay? 1, 2, 3, 4, 5, 6 from the end, that's an omega-6 fatty acid. And it's really these omega-3s that we care about. And when you care about the ratio of omega-3 to omega-6 fatty acids that you're consuming in your diet, if you're somebody who's really highly interested in monitoring your nutrition, the omega-3s are fatty acids. They're PUFAs, they're polyunsaturated fatty acids. They always have this double bond at the 3rd carbon atom from the end of the carbon chain. And we found that they have certain properties that we believe are healthier, okay? You'll find more of the oils that we use have this 6 omega structure, and that's okay, but we want to get the ratio so that you're actually getting more. You get more of this stuff from, like, if you eat, you know, salmon, or in other deep sea fish, some nut oils are omega-3, some really good for you.
Fish oils as well.
Yeah, fish oils, but you got to watch which fish oils.
Right.
Like, you know, when
I
they
got a
try to
quick
sell you all
question.
the krill oil and all that type of stuff. So,
I got a quick question. Can you hear me?
yep.
What, this is probably a little off topic. So what are the benefits of having the double bond at the 3rd carbon versus having the double bond at the 6th carbon? Is it
Yeah.
about how the fatty acids can stack up on top of each other?
Yeah, we'll go through this a little bit more later on. This is just an intro. When we get into our nutrition topics, we'll talk about it, but
Okay.
I anticipated a little bit of chat about this. So I just threw in this slide last night of, you know, what is all the fuss about omega-3. So you find it in fish oil, flaxseed oil, even hemp oil supplements, okay? Now, the thing is, they thought for the longest time that consuming these oils was going to be associated with a lower risk of all-cause mortality, but it's not doing that, okay? That hasn't panned out. Where it is significant is with cardiovascular disease. So if you're consuming greater than 1 gram daily for at least a year, it appears to be protective against cardiac death, sudden cardiac death, myocardial infarction in people who already have a history of cardiovascular disease. It may reduce the risk of stroke and blood pressure in people, and it helps reduce the overall level of blood triglycerides. The consumption of the stuff of omega-3s from marine sources does seem to help with inflammatory markers in the cell, okay? Lowering all those things that get cranked out when we have inflammation, like C-reactive protein, interleukin-6, TNF-alpha. These might be names of substances that don't mean a lot to you, but they will once we get into our immune section, okay? And so it's uncertain if any of this stuff, omega-3s working on the inflammatory system is actually all that useful, okay? So we're kind of just touching on it light. What is an omega-3? When we get down into the heart and cholesterol and how we're utilizing our fat in our nutrition, when we get to those chapters, we'll talk about it a little bit more. Right now, we're learning the molecules, okay? All right, so when we get into complex lipids, fatty acids were simple. They're the long chains, and we've just joined them on to glycerol to make this more steady and more complicated triglyceride molecule. But it's not the only complicated fatty molecule that we have. We have other complex lipids, and these two will contain carbon, hydrogen, oxygen, phosphorus, nitrogen, and sulfur. All right, so we all know that our cell membranes are made of the complex lipids that we call the phospholipids, right? And our phospholipids will look a lot like those fatty acids we just built, where they'll have the involvement of glycerol. And instead of 3 fatty acids, they'll be attached to 2 fatty acids and a phosphate group. Phospholipids will have a polar region as well as non-polar region. You can already predict that the fatty acid region is going to be the non-polar region, right? And that this phospholipid head up here is going to be the polar region. All right, so let's look at our phospholipid heads. Try to get this out of the way. Looks like he's walking down, doesn't it? All right, so here is your glycerol molecule right here, the 3 carbons, right? And here's kind of attaching the fatty acids the way they normally do. On your 3rd carbon is where that phosphate attaches. And that phosphate attaches and orients itself up and away on the other side, kind of in a trans-like formation from the direction the fatty acids are going to attach to the glycerol molecule. So you can have completely saturated fatty acids on here, or you can have, as in this case, we see a fatty acid that has the bend in it right here, an unsaturated fatty acid. Whoops. Okay, so the long tails of fatty acids these are going to be our long hydrophobic, right? These are going to exclude water. This head with this phosphate, which is really charged, you see the one charged up here, but really in practice, you're gonna always see this kind of, these electrons kind of are more sticking around loosely here on this. It's all very negatively charged organic head of the phosphate group. So these will line up in a row. Phosphate heads with their charges, negative charges, and all this is going to be uncharged. Uncharged in the center. So uncharged in the center is going to exclude water passing through. Exclude, it looks like exude, exclude water. Okay, in here. But these charged heads, they want to face water. Okay, so they'll face water. Out here, let's say this is going to face the water in the extracellular environment. And if this were the inside of the cell, it would face the water on the inside of the cell. Here we can see if you had fatty acids that had a lot of kinks in it, still even the little tails or legs on the fatty acids, they're a little bit more open than that, but they're still going to be excluding out the water and any polar substances. They're just a little bit more loosely packed in here. And then you see when we get the whole fluid mosaic model going, we see our fatty acids all lining up, and interspersed in between, we're going to have some cholesterol molecules stuck in amongst our fatty acids, okay? Those cholesterol molecules will give both some regular rigidity structure to the molecule, and at the same time also make the membrane bend and be more flexible and have a little bit more fluidity, which is hard to understand how it can give some structure or rigidity to the membrane and fluidity at the same time, but it does. All right, maybe
Good question.
Start making more sense. Yes.
Will it always be a combination and with a one saturated part of the phospholipid? One leg will be a saturated, one will be an unsaturated, or it can,
No,
it's a variety.
no, it can be anything. Yeah, you can have, you can have all completely saturated. You can have two unsaturated. You can have a saturated and an unsaturated. It's just however that individual fatty acid is made.
And does that dictate what type of things it will let permeate through? Does that affect it? Is there any correlation, or is that getting
not so much
down the wormhole?
Not so much. You're going down the wormhole on that of thinking that would make sense. It's just kind of random. They just pick up whichever fatty acids they have to make themselves available. And, there's so many fatty acids crammed in there that you can get some that have this unsaturated fatty acid, and it'll be crammed in with so many saturated fatty acids around it. But the whole thing becomes a water exclusive unit. All these tails in here facing into each other, it makes a really good structure that keeps water out. And we'll see that on the next slide. So when you have large numbers of fatty acids that have these hydrophilic heads that have the little charges on them, whichever way you want to draw it, and you're going to have your hydrophobic tails, whether they're having a cis or a trans bend in them, or whether they don't have any bend at all. All these will kind of be crammed in there, one next to the other, and you'll see that they line up, and on their own, how they form to make the cells really is they form micelles on their own. So the micelles will have the fatty acid heads facing the exterior of the aqueous environment. And then all the tails line up in the center to be exclusive, and they make that kind of almost compact droplet or ball, okay? If left on their own, they kind of like bead up. And then you can see how it's got a couple of sort of little sugar moieties kind of sticking up here that might be able to interact with the water. But this is just a core of a big giant lipid droplet. These are like the lipid droplets that form in your salad dressing. Now, you know, in your cell, of course, it's going to end up stretching out and panning out and making a membrane, and it's going to have a lot of things like lipids and that holding it so it doesn't bend and round up just to make a ball, okay? It's going to form the whole outside area of your cell. So we call it, these molecules amphiphathic in nature, okay? Meaning that the fatty acids have both these hydrophilic regions, okay, that are polar and attracted to water, but as well, they have the regions that exclude water, right? All this down here, all this non-polar. So there are two things. They're amphiphathic. They have both polar and non-polar in the molecule. So one end allows of the molecule to interact with the aqueous environment, but this big long end here, of which we will have two facing each other in our cell membrane, so it makes it quite thick, of non-polar. Okay, so that's going to exclude all that water that you have there, all that long hydrophobic chain. All right, moving on, our next liquid that we're concerned with, I shouldn't say lipid, that we're concerned with are steroids. Okay, so steroids will all be based off of this 4-carbon ring and 3 6-carbon rings that are somewhat open and 1 5-carbon ring. Steroids will be placed into our growing membrane structure, and they both give the membrane some rigidity or structure, but also give it fluidity, where you're breaking up some of that, you know, hydrocarbon chain, fatty phospholipid, hydrocarbon chain, phospholipid, to make things a little bit more fluid through the membrane. Okay, so you'll recognize this nucleus right here with this A, B, C, D, with this being the 5-carbon out here. Okay. So when we look here, here's again that steroid sort of basic structure. Here is cholesterol with your A, B, C, D, D being the 5-carbon structure. And you'll just get used to recognizing it, okay? And it has different substitutions off of some of its chain. So we've talked about with lipids, we've talked about the generalized fatty acids. The generalized fatty acids get attached to the triglycerides, okay? And these are going to make the basic structure of every cell wall membrane, okay? Every cell wall membrane. And now we're looking at cholesterol, okay? And the steroids. We derive all of our steroid hormones from cholesterol. Cholesterol is a major component of every cell wall in your body, okay? And it confers both some level of rigidity, but also fluidity to all the plasma cell membranes. So these are large lipid molecules with 4 major carbon rings, that A, B, C, D ring, A, B, C all being 6 carbon rings, and the D ring being a 5 carbon ring. And they differ in their structure by the functional groups that are put on them. Okay, so cholesterol, remember always, component of cell membranes, absolutely necessary for life. Okay, necessary for life. No cholesterol, no life. Doesn't want to write here. It gets kind of a bad name, right? From cholesterol, we'll make our estrogens and testosterone. When you look at estrogen and testosterone, you can see how they're derived from cholesterol. We'll make our corticosteroids. Yes, all those things that are anti-inflammatory. Calcitriol for our vitamin D. The bile salts are also made from cholesterol. Your bile salts are stored in your bile duct, right? They're made in your liver, and they work with the lipase enzymes, and they'll help sort of solvate or digest the lipids in your intestines so you can absorb dietary fat. All right, so dietary cholesterol, I just said it's a necessary component of every cell membrane of your entire body. All right. Why does it have such a bad name? And a lot of its bad name comes from old thinking about cholesterol. You know, they would see the cholesterol in the people who'd had heart attacks. They would see the cholesterol maybe as part of the fat blockage and components in the blocked arteries. And they thought the cholesterol was the culprit. But, you know, the more and more we study cholesterol, we know that we need cholesterol. Like I said, it's a functional part of every membrane. It's the base from which your hormones are made. We only get 25% of our cholesterol from our diet. 75% is produced by our body. It's made by our livers, okay? So if you're getting 75% of your body producing it to begin with and only 25% from your diet, do you think dietary cholesterol is really the evil source of problems in our diet? No, probably not, right? Okay. And this is why things have switched around for treating cholesterol is not so much telling you to control the cholesterol in your diet as it is trying to give everybody cholesterol medication to control the amount produced by the liver. Your body tightly regulates how much cholesterol it's going to make, regulates the amount of cholesterol that's flowing around in your blood. This is all part of homeostasis. And so if your dietary intake of cholesterol goes up, you know, the body's going to make less. If your dietary intake of cholesterol goes down, your body's going to crank out more endogenous cholesterol. And, you know, there are some people who genetically just make too much cholesterol. And that's why we have all these wonderful anti-cholesterol drugs of different classes. All right. We'll talk more about cholesterol later on too when we get into our macromolecules and nutrition. Okay, so proteins. Proteins are made of carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. And they're essential for our cell structures. They, you know, make up our structure, okay? And for some functions. For example, enzymes are proteins that are functional. They help us, they're catalysts that help us speed up reactions. We also have proteins that are transporter proteins that will move chemicals across membrane. The flagella on sperm is a protein. And some of the bacteria that live within our body are also using their proteins to make toxins in their cell structures. And some of these bacteria are good microbiota. We need them. All right, so proteins, their monomeric unit, or what they're made up out of, are going to be the subunits called amino acids. And amino acids will be structured such that they have an alpha carbon right here. I drew the arrow down to it. We could write a little alpha. Whoops. I have to go back up here when I get the draw. Okay, and we could call this the alpha carbon right here. Okay, the alpha carbon. And around the alpha carbon, we see attached to it is we have an amino group and a carboxyl group. So get that again, here's our carboxyl group. Let's get our amino group in blue. Okay, and our R group down here. The R side chain could be anything. It's usually something, you know, like a long group of hydrocarbons all ending. You know, you could have it saturated with hydrogens. Something like that, okay? So we just always write R, okay? Long chains of hydrocarbons. Your amino group is going to be your NH2. Okay, when you have your amino group, you also know that your nitrogen has its lone pair of electrons. Now, what do we care about all this structure for? It's always going to be amino group, carboxyl group, R group. We try to line them up there. Here's that R group being some big carbon group here. And what you're going to find is that we're going to form a bond between this amino group and this carboxyl group to put them together to start to make polypeptides, a peptide bond, okay? So we're going to have the OH and the H. We're going to have an attack like the, let's say the two dots over here, the electrons might come in and attack this carbon, and it's going to boot off this water, okay? And what it'll do then is form a peptide bond between the nitrogen and the carbon, so that you would end up seeing, you have this NH2 out here. You have this carbon, that's alpha with its R on it and its H. We have this carbon right here is now going to be bonded to this nitrogen over here with its H to another alpha carbon, which is then bound to a COOH. Okay, this alpha carbon, I should probably make it look more like an alpha than a 2, has that big R group, that cyclic side group, and this has this cyclic group on it. This right here is going to be the peptide bond, and we got rid of this hydrogen and this hydroxyl group, so we got rid of water. So we synthesized it and got rid of water, so it's a dehydration synthesis or a condensation reaction when we made that. Same thing happens over here. Here you can identify, we have our amino group, okay? Here we can identify, you have your carboxyl group. This right here is your alpha carbon, and you have the R group hanging off of it. Again, your carboxyl group, your amino group. This is the alpha carbon. Amino group. Carboxyl group, okay? And it's always going to be carboxyl group attacks this amino group, or amino group attacks this carboxyl group, probably. Like, got lone electron pairs, decides it's going to come and attack this carbon, right? When it attacks this carbon, it's going to remove off the OH here and this. We're going to boot off our water. We're going to condense. And then we're going to have our new bond form there. Okay. Here's all the amino acids found in proteins. And so, you know, it's just kind of getting to know them. You don't need to memorize them. The simplest one is glycine. So if your amino acid group is always a carboxyl group, COOH, and an ammonia group, NH2, you have your carbon, alpha carbon. You're going to have an H up here. The simplest R group is just an H. Alanine is the next simplest. Here's your amino group. Here's your carboxyl group. Here's your alpha carbon right in the center, always an H. And the next simplest thing that you can put on here as the R group is a methyl group, CH3. And so they just get progressively a little bit bigger and more complicated as you go along the path here of looking at all the different amino acids. All right. Okay, your amino acids can exist in either of two stereoisomeric forms, D or L. This is probably getting a little bit more complicated than we need to be, but your book goes through it, so we shall. The L forms are more often found in nature. Think of natural, and they all have the L image. So what is a stereoisomer? Stereoisomers are mirror images of each other. So if this guy over here, this amino acid, because it's a amino acid up here, and amino right here, R group H, and here's your alpha carbon, looks in the mirror, is going to see the exact same thing. Just like if you put your right hand and your left hand up to a mirror, it's going to look like the exact same thing. But you could never take your hand and superimpose it on its mirror image. That's the same thing here. If I move NH2, it's going to be right here on top of the H. And the H is going to be right here on top of the NH2. So it has a specific orientation in space, and that affects the molecules that it interacts with, can only have the specific orientation in space that aligns with that stereoisomer. Okay. All right, so that's a little bit of a side. It's not as important as understanding the peptide bond. So going back here to our peptide bond, being able to recognize the amino end of the amino acid. Remember, it's called amino acid because here's the amino group. It looks like ammonia, amino, and here is the acid group. Okay. This is the alpha carbon in the center. All right. we're going to have one amino acid right here of glycine is going to interact with this amino acid of alanine. Here is its COOH or acid group, and here is its amino group, right? And we know that the nitrogen is going to go with its heavy electron load there, is going to go and attack the bond over here and kick off that water, right? In a dehydration synthesis. So all this water is now left. And we now have a new bond formed by that dehydration synthesis. So you'll see the new peptide bond right here. Single bond drawn in, okay? And here's the water that was kicked away. And it goes in the same order. NH2, CH2, CH2, CO, CO, remember the OH was removed. And then we have the new bond, new peptide bond. We got rid of one of the H's, so now we're just NH, so NH. And then we have our other alpha carbon with a methyl group on it, and then COOH. We removed water and made that strong peptide bond. And we could do this over and over and over again. And instead of having a dipeptide and a polypeptide, eventually you would have a whole protein. All right, so proteins. We now look at how we take our amino acids and another amino acid, and we get the basic structure of one amino acid after another, after another. That's called the primary structure of the protein, the simple order of amino acids as they're strung together like pearls on a necklace. Okay, it's simply the sequence order. Alanine, glycine, alanine, glycine, phenylalanine, you know, going on and on. All right. The secondary structure is that once you start getting all of these primary structures together, they start to take on, because of hydrogen bonding between the molecules. Remember, hydrogen bonds are more like loose associations, but they kind of like associate in specific folding over on each other manners. And they can fold over as pleated sheets. They can twist up as alpha helices. So that's their secondary structure. The tertiary structure is after they've made, say, like an alpha helix twisting up, okay, is that then they might also even kind of fold back on each other. And you see here, here's twisted, 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 alpha helix, and then those twist and fold up on each other into a third level of structure, that tertiary structure. And then sometimes we'll have one long protein chain that's all twisted and folded up in tertiary structure. And we'll have another one that's all twisted and folded up in its tertiary structure. And they kind of come together, and like one might fit in a little nook on another one. And that's its quaternary structure, okay? That's its quaternary structure. We'll see also that amongst all these proteins, the proteins can undergo a process of denaturation, meaning maybe we can heat it up and start breaking some of the bonds, okay? Start changing the shape, all these nice conformations that this protein made. We could put it in a high pH and it kind of will change and maybe break apart some of the hydrogen bonds that kept it, you know, folded in a specific way. All kinds of things that you can do to denaturate a protein. When you're getting a permanent and you want to get permanent curls put in your hair and you get your hair all wrapped around, somebody comes and puts chemicals on the proteins on your hair to denaturate the proteins and make it stay in the curl that you put on your hair, okay? Conjugated proteins consist of amino acids and other organic molecules. So sometimes our proteins might have a little sugar moiety or a little piece of sugar added on to them. Okay, maybe they have a little piece of lipid added on to them. You know, if they have sugar added on to them, we could call it a glycoprotein. Or if the sugar is the primary component and we added some protein to it, it might be more like a proteoglycan. Okay, so nucleoproteins, proteins that have a little piece of nucleic acid added to it, and so forth. All right, so let's talk about nucleic acids. Nucleic acids are made of nucleotides. And what are nucleotides then, you say? Nucleotides are made up of a 5-carbon sugar, a phosphate group, and a nitrogen-containing base, all right? When you have just this sugar and the nitrogen-containing base by itself, we also call that a nucleoside. So let's start looking at some of these. Typically, when we're wanting to build up these nucleotides, the thing that we're interested in ultimately is looking at DNA, deoxyribonucleic acid. It's going to contain a sugar that's called deoxyribose, all right? And we know that its shape it likes to take is the double helix. And so we find in these base pairs, we're going to have bases that regularly pair with each other. Adenine will bond to thymine. Okay, adenine to thymine, and cytosine will bond to guanine. Okay, all right. The order of all these bases going down the middle of our DNA molecule is kind of the code, the secret code that forms the genetic instructions that tells the organism how to make, you know, contains like the recipe for making all the proteins. It's kind of the genetic basis for making every individual organism. The genetic information in bacteria and a lot of our viruses will also be encoded in DNA, although we'll see that some viruses use RNA. All right, so here's the chemical structure of DNA. These are these base pairs going in here, one after the other. All right, so we had a guanine here and a guanine here. Guanine only pairs with cytosine, so that's C and G. If this is thymine here, this is adenosine over there, okay? So adenosine, thymine. A pairs with T and C pairs with G. A will form like 2 hydrogen bonds with T, and C will always form 3 hydrogen bonds with G's. And you can see that over here. You see the hydrogen bonds going between the H and the O there, between the H and the N right here, okay? That's what's holding your molecules of DNA together in these long alpha helices. Isn't that crazy? But it's like what I was saying. They're loose associations. They can, you know, easily decompose, break apart. But in sum total, when there's so many of them, just like keeping all the water molecules together in a beaker of water, they add up, right? Okay, they really add up. But you can see why you want something that's a little bit more fleeting of an association with your DNA, because as you've learned before in high school, we're going to need to unzip that DNA, right? We're going to need to open it up and read this genetic code, okay? And if we had all these covalent bonds or some really strong ionic bonds in here, holding these two halves of the alpha helix together, they wouldn't come apart easily enough for all our needs to keep unzipping and rezipping and unzipping and rezipping and unzipping and replicating, you know, unzipping and replicating, unzipping and transcribing, okay? You wouldn't be able to do it if we had all those covalent bonds, okay? So when we look at DNA, remember, you're going to have going all down the backbone here, that's going to be that sugar phosphate base, okay? So all those sugars, all that deoxyribose going down the bases. The rungs of the ladder are going to be all of the nucleotides, okay? And those nucleotides are going to be held together by these hydrogen bonds, okay? We can then have our DNA open up when we get something like a DNA polymerase on it, right? And the DNA can get read. And when we read it, we transcribe this length of DNA from DNA to RNA. That's transcription. And then our RNA is what leaves the nucleus, right? For translation. This leaves the nucleus and goes out into the cytoplasm where it meets up with the ribosomes, and the ribosomes will read the secret code and bring transfer RNA over to translate the secret message here into proteins, okay? All right, we'll keep trying to put it together more and more. All right, so RNA, instead of deoxyribonucleic acid, RNA is ribonucleic acid containing ribose. It is single-stranded. And when we had DNA, we had adenine bonding with thymine. We now have adenine bonding with uracil, okay? We still have cytosine forming its 3 hydrogen bonds with guanine. I can write that back correctly. All right. Now we're going to have lots of different types of RNA, and they're all going to play a role in protein synthesis. Yep, Saba?
Okay, so the DNA has those 4 groups, and the RNA is running the same situation as the DNA, like with the group to make the protein?
No, so DNA compared to RNA, DNA is going to have adenine and it's going to bond with thymine, cytosine, and guanine, okay?
Mm-hmm.
This is always going to stay in the nucleus. It never leaves the nucleus, all right? Now, RNA, instead of adenine bonding with thymine, we now have adenine bonding with uracil, okay? All right. DNA is double-stranded and opens up. I'm trying to write this so that it's somewhat neat. D-O-U-B-L, double strand. RNA is single-stranded. Okay. All right, now RNA that we're going to do here, this leaves the nucleus, DNA is always going to stay in the nucleus, but this stuff gets made in the nucleus, okay, it gets transcribed off of DNA, and we'll go through that, but it's going to leave the nucleus because it's what's going to take the message on a single strand over to the ribosomes. And the ribosomes will use the order of all these nucleotides to tell what protein to manufacture. Okay. All right. We'll keep going through this and hopefully it'll start making sense. All right. So here, when we look just individually at a nucleotide of RNA, a nucleotide is going to have a phosphate group. Okay. So lots of negative charges end up being on the phosphate group. This is why this can face out toward water. You have the ribose group, the sugar moiety, okay? And then up here, you have your nitrogenous base, okay? So that forms the uracil. Now, if we also want to look at not just DNA and RNA, we also need to look at the big molecule of ATP, adenosine triphosphate, because it too is made of these nitrogenous base groups and the sugar groups. So they're similar in their structure. Adenosine triphosphate, you'll see, is made of ribose down here. That should look familiar. Okay. Adenine. Okay. That should look familiar. And then it has 3 phosphate groups. Sometimes they're written like PI, and PI for abbreviations. Okay. Contained within all of these bonds on here, this bond right there and this bond right here. When we strip one of these bonds, it is full of energy. Okay, and that energy can be used to drive all kinds of reactions in the body. So this is where ATP becomes the currency and becomes so important in the body, is that when a reaction needs some energy to drive it, like an enzyme maybe building up a sugar or a carbohydrate or something like that, making something the body really needs, we might have to use the energy in this bond, and so we'll split this off. Okay, when we split this off, the rest of the molecule over here becomes ADP. All right, and all the energy that was in this bond, we're going to use to drive other reactions. Okay, so we capture and use energy in the high energy.
So what does the squiggly line indicate when we're looking
These
at the
squiggly lines
structure?
right here, like this?
Yes.
These are high energy bonds. Okay.
Is
These
that
are
like
the
a
bonds.
universal symbol for
Yeah.
high energy bond in
Yep.
chemistry? Okay.
Yep. High
Thank you.
energy bond. Okay. And that's the one where you split it off. Okay. And so you can use that energy to drive reactions that might not otherwise go, that need all that energy being put into it. Typically anabolic reactions, things that you want to build up in your body and you need all that energy put into it. All right, so let's talk about that, ATP. So ATP stores chemical energy that has been released by other chemical reactions. And when we release the phosphate groups by hydrolyzing them, we liberate useful energy for the cell to use. The energy is stored in phosphate bonds, and those are those high energy phosphate bonds, kind of written squiggly like that. So you always see how ATP is considered the currency of the cell, kind of like that you can store energy in the molecule, and you can break off little pieces of that phosphate when you need to pay and use some energy, okay? It's kind of like the bank of all your energy. So here you see it, just instead of drawing out all these phosphate, you can see that it's written as adenosine PPP, okay? And so breaking this first phosphate off here with water would release more energy. Breaking off this next phosphate bond, you'd release some good amount of energy, but not as much as what's stored in the first bond, okay? That's typically the way it goes. So if you broke this bond, you would then have ADP, adenosine diphosphate. If you went further and broke this bond, you'd have AMP, adenosine monophosphate, okay? And I have some things here about this Cassiopeia project that can help you with understanding some of this, and as does the Khan Academy videos. And I have more videos, but we're going to go into right now, chapter 4, the cell, which kind of picks up with all of what we've been doing, okay? It's still a lot of the cellular energy stuff. All right, hang on. Make sure I get on to share. I know this is really, really hard stuff. Can you all see this? Cellular metabolism?
I
Okay.
can see it.
Okay. I can see it and I don't like it, right? All right. Okay, so we're going to start trying to put it together a little bit, some of these macromolecules and what we care about. All right, so the idea is that all your cells are going to undergo metabolism. And metabolism has two big main parts. Anabolism, which are going to be the synthetic reactions. This is building things up, going from simple molecules to much more complex molecules. Protein synthesis is a central anabolic pathway building up these programs, building up these proteins. So this is going to require energy to be input into these molecules. You'll probably get a lot of that energy from ATP to make the molecules that you're making in anabolism. Catabolism are the decomposition reactions, okay? This is breaking down complicated molecules into simple ones, and these will release energy. That energy can get stored and used later. So catabolism, eating the donut that you had for breakfast, breaking apart all those sugar, polymers of sugars, all the starch, the simple starches, the simple carbohydrates, down into their monomeric sugar units, so that the little itty bitty sugar molecules can then get absorbed at the brush border of your intestine and taken up by your body. And then those sugar molecules will get transported around in the blood. Some of them might go to the liver, where we'll kind of actually build them up again and store some of it for long-term storage as glycogen for when you need, you know, some sugar release later on. Some will go directly to the cells, okay? So that's kind of how the fate of your sugar donut breakfast goes. Trying to piece it together and make us think. All right, so cellular metabolism is going to be the sum of all the chemical reactions that are occurring in the cell. You're going to have some anabolic and catabolic reactions all going on at the same time. And we'll see that these reactions tend to occur in pathways or cycles. So what we see here, we have a monosaccharide and another monosaccharide And over here, it looks like we've joined them to make a disaccharide. We do this, as we just have learned, by removing water, right? We do condensation reactions. Our condensation reactions are also called dehydration synthesis reactions. Okay, so we're going to remove water right here, and we're going to form a bond right here. Actually form an ester linkage. Okay, so this is dehydration synthesis in this direction. All right. Now, if we had this disaccharide and we needed to extract the energy from it, and we wanted to break it apart, we would use water to attack that bond and break it apart. And this is called hydrolysis. This is the reverse of dehydration synthesis. Okay, and it breaks that bond, and then you end up with your sugar that I'm not drawing very well. Okay, with this H, and now it has an OH on it, and you're going to end up with your, boy, am I really not drawing sugars well. Okay.
So is all metabolism processes either dehydration or hydrolysis? Is that a correct statement?
They're not all, but pretty much our major ones are. All the ones that you need to learn for this semester are dehydration synthesis, which are also called condensation, because you're condensing or taking water away, like sweetened condensed fruit juice in a can, right? And you can reconstitute it by putting the water back in, okay? All right, so dehydration synthesis means we're always going to form bigger molecules from smaller reactants by removing water, okay? They're a subset of a larger group of condensation reactions where you remove water in order to form a new bond, okay? Anabolism, okay? Anabolism, building things up, getting stronger, right? Anabolic steroids. Anabolism is we're going to provide all the materials to maintain our body, okay? And give us also all those nice substances that help us grow our cells and repair our cells. And it's going to require ATP. We're going to have to put energy into the system, okay? All that ATP we extracted out of molecules during catabolism, we can now use to build things. So you always have to think, catabolism, you know, you ate stuff, you're breaking it apart down into simpler things, you're extracting all the energy out of it. But at the same time that you're eating and extracting the energy out of your foodstuffs, you're also trying to put energy into molecules and build all those structural molecules like your big, strong muscles, you know, maintaining your bone strength, repairing like the little hole you got when your dog scratched your face or something. All that stuff is going to be anabolic, building you back up, repairing you, okay? And all the energy, all the particles, all the foodstuffs, that eventually comes from basically what you ate, okay? All right, so examples for anything anabolic is we're going to also do dehydration synthesis or condensations to build our anabolic molecules. So we're going to take the two smaller molecules, like the saccharides we just broke apart from your donut that you had for breakfast, okay? And we absorbed them. We had to break them down in our gut, and now we've got them all nice and small, and we had to absorb them at the brush border of our intestine, right? So it gets taken up into our blood, and now it has to travel somewhere in our body where our body needs these sugars for something. And let's say we're just going to store it, build it up into disaccharides and glycogen in the liver. So maybe all this glucose that you ate this morning travels over to your liver. We have one monosaccharide, two monosaccharide, and they're going to undergo condensation, dehydration, synthesis, condensation in this direction, right? And a new bond forms between them. So now they are a bigger molecule storing more energy, particularly in this new bond. And we have water removed, water as our side product, okay? Again, dehydration, synthesis, condensation reaction, same thing, okay? Just two different words for the same thing. In this case, you're combining smaller molecules and you're building them into a bigger macromolecule, and you extract water to do it. So right here are two sugars, glucose and fructose. They're going to get combined into a whole new bond here. And water's what's going to get removed. So water's removed. Pretty typically, when you're removing water, you see it getting like dropped out as the arrow. And then you see you have your new bond from this carbon over here to this carbon over here with the oxygen in the middle. It's an ester bond, COC, okay, is sucrose. All right, so when we look at anabolism and catabolism of fats and proteins, okay, so here's back to our friends. Here's glycerol. Every carbon looks the same. CH, CH, COH, CH, COH, CH, CH, COH. Okay, so you can see where part of the water is going to get removed off on glycerol as it attaches to fatty acids. So here we have a 1, 14, 16 chain carbon that's going to attach up here. Same thing, 16 chain carbon going to attach, doesn't show any double bonds on any of these. So it's going to be the same 16 chain carbons, dehydration, synthesis. You can see we're going to extract a water and a water and a water. And we're going to have our new bond being formed from this carbon over to this carbon, right? But there's going to be an oxygen still in between. So it's an ester linkage. Okay. This oxygen is staying right here each time. All right, so estrogen, ester linkage, and then 3 molecules of water removed. If we wanted to do the reverse and break apart, this is now a big triglyceride. Triglyceride, meaning here's the glycerin and here's the 3 fatty acids off of it, triglyceride. If we wanted to break it apart, we'd be breaking apart this bond right in here that we just made by having all this water attack these bonds, okay? And the reverse would be hydrolysis. I just kind of crossed right through it, okay? And we break off all those fatty acids and be left with glycerol. All right, here you see the anabolism and catabolism of a fat, up there, fat, and then an amino acid. So we have our amino group, we have our carboxyl group. And it's between the carboxyl group of one amino acid attacks the amino group of the next amino acid. Okay, so this goes in and is going to sort of look over there at that nucleus and say, I want to form a bond, and all this water will be kicked out. And here you have it, the same amino group. You have your carboxyl group, but no longer the OH attached to it. You have that bond, the bond right there. That's this bond over here. Okay, this H went with the water, right? So we just have one H left on the amino group down here. And then you still have the C with the R, and it has its acid end. So now you have a dipeptide. You have two amino acids together. That bond is called a peptide bond. Okay, now catabolism, breaking down the bigger molecules into smaller one. ATP is usually produced. Okay. We often use hydrolysis. We use water to break apart the bonds of carbohydrates, proteins, and lipids. You use water to split these substances apart. It's the exact reverse of what we've been doing with the dehydration synthesis. So we use water, like I said, it's going to come in here. It's going to attack the bond, all right, from our disaccharide. And we're going to end up with putting hydrogens and oxygens on here. This hydrogen and oxygen comes over here as H and then O, and then we put our other H. So you see HOH over here, HOH over here. All right, we've just split it apart. Catabolism of our lipids, same thing. All this water here is going to attack all those bonds that we just made and add hydrogens onto the oxygen over here. All right, same thing. Water attacks our peptide bond, and we go in reverse for hydrolysis. And we get back our two original amino acids. So you have all of these metabolic reactions. Your cell has ongoing performance of both catabolic and anabolic reactions happening in your body all at the same time. But the rates of these reactions have to be carefully controlled. This is all part of homeostasis. You can't just be willy-nilly breaking down all the structural proteins of your body. We'd all be Gumby, right? We'd have no, structure. All right. So the breakdown energy releasing reactions have to occur and be balanced with the building up. And when you get way out of whack, you also can find you can have gaining weight, gaining muscle, when you're not in balance with that. Imbalances in your reaction rates can kill a cell. Okay, so this is why that homeostatic mechanisms are really important. The cell is very fine-tuned in needing all these reactions happening just right in order to, keep its homeostatic base going. All right. Different cells will undergo different metabolic reactions. Not all cells are, say, making, aren't collagen factories. Certain cells will make a lot of collagen. Not all cells, but some cells, particularly your pancreatic islet cells, will be making lots and lots and lots of insulin. Now, of course, all your cells will have the genes in them that have those instructions for making insulin, but it's really only the specialized cell for making insulin that makes insulin to any degree, okay? Your epithelial cells might manufacture melanin, okay? But your muscle cells, for instance, aren't going to be doing that. They're going to be busy, making the proteins that are needed for the muscle, okay? Like actin and myosin. So every different cell will have its different focus of metabolic products that it's trying to make, all right? And it's going to be enzymes that will control how much is being made and what gets turned on, kind of turning on the genes that say make insulin, or turn on the genes that say make melanin, all right? And those enzymes will then, function to really enhance the rate at which the product is being made. All right, so let's talk a little bit about enzymes. Enzymes are protein catalysts, okay? They are globular proteins and they catalyze very specific reactions. They will increase the rates of chemical reactions, and they do so by lowering the activation energy that's necessary to start a reaction. Enzymes are not consumed in the reaction, okay? They just participate and then they kind of go off away so they can participate in other reactions. They just kind of help really bring product, two reactants together to make a product, all right? So they're not consumed, they're used repeatedly. Each enzyme is very specific to a particular substrate. It's going to have the ability to recognize a substrate based on the shape of that protein of the substrate, and it's going to fit right into its active site, all right, in order to activate it. And kind of a lock and key, it has to be just exactly precise fit. Many of our enzymes are named after the substrate that they utilize, and then they're given a little phonetic ending called ACE as its suffix. So lipase fits into a fat and helps act on the fat, breaking it down, lipase, okay? Sucrase would act on the sucrose sugar, and so forth. All right. So when we talk about the control of enzyme reactions and how they promote the lowering of the activation energy, you probably have somewhat learned this in chemistry before and forgot about it. Okay, the activation energy is the energy barrier that a reaction needs to get over, this hump, in order to get the reaction going, in order to get this party started, right? Okay, in the words of Pink. Enzymes are going to be these proteins that they're made by the cell, and somehow, usually they'll work by maybe like holding the two reactants actually close together to make the reaction more likely to take place. All right, so a reaction without an enzyme, all right, means we start out with the same reactant, okay? Here's this AB stuff. And without using a catalyst, it's going to take a boatload of energy. You have to put more and more free energy into it to get this reaction up to the top of this hill, for it to roll down the hill here and make the two products A and B, all right? Now, with an enzyme can somehow hold that product such that it takes a lot less energy. This hump that this AB has to get over so that it can become A plus B is a lot lower, a lot reduced, okay?
Just like a coupon.
Yeah,
I'm
kind of like a coupon, or, it's knocked off
very
all this
specific
up
to a
here.
product.
You just don't have to get up that hill. Okay. And so then it makes it that much easier to get to the products. All right. So instead of a coupon, I always liken it to Sisyphus. You see, I like Greek mythology. And Sisyphus was in Greek mythology, the guy who's always trying to roll that big giant rock up the hill. And the closer and closer he gets up to the top of the hill, the more likely it is that he's going to start rolling right back down repeatedly over and over again. All right. Now, so I always say the chemical reaction seems like a Sisyphean task, trying to get up over this big giant hill, and it's like you're going to push and push and push and likely fall back down and still end up where you started. But the enzyme comes in, and an enzyme is like a bulldozer, okay? And it's going to plow down that energy hill and make it a whole lot easier for you to get over that hill. So here you are with your reactants trying to get up over that hill, that Sisyphean task, and you can't do it, but along comes your enzyme with its bulldozer, and it plowed down this hill all the way down here. So suddenly your reactants can easily go to form stable products. All right. So that's with an enzyme. All right. Makes that progress of the reaction that much more likely to occur, that much easier. All right. And then we'll get into our enzymes fitting in lock and key method as protein catalyst. We'll pick up from here next time. I'm going to go jump in the car and come out to Ann Arbor and see all of you who are coming out today. Like I have said, any of you who are scared that you're going to get snowed out and not come on Thursday, if you want to come and join us today on Tuesday, you're certainly welcome to do so. Those of you who aren't even going to be in a lab this week because you've been sick, COVID positive or whatever, and you're worried about the quiz, it's not a very big quiz. You can take that quiz the next time we all meet together in person, okay? And that's it. I have to run off, so